Awesome. Cool. Beth, are you ready with the intro?
I am ready. Are you ready?
I'm ready.
Hello, 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 and welcome to the Smooth Brain Society. Now, what do you get if you cross a neuroscientist, an engineer, a comedian, and someone who really likes shining lights into brains? You get Liam Collins Jones, and that is the guest on our today on our podcast. Now, Liam's building the next generation of wearable brain scanners at the University of Cambridge, shining infrared light through people's skulls to watch their neurons fire in real time. He started out strapping tiny light caps onto babies to figure out how their little brains work, and then moved on to mapping how hearing loss messes with your brain as you age. Now, apparently being in the lab developing cutting-edge imaging techniques just wasn't quite enough for him. He now also performs as part of Three Queers and a Welshman, making people laugh about science. Now he's performed at Glastonbury and Green Man Festival, done time at the BBC, and convinced people that neuroimaging and the brain can be pretty funny. Today we're getting into the nitty-gritty of brain imaging techniques, the surprising link between hearing and cognition, and why making people laugh might be one of the best ways to explain your research. Welcome, Liam.
Woo! Thanks, Beth. Happy to be here.
Glad to have you. So to start us off with, can we ask you, what is your origin story? How did you get to where you are today?
Well, firstly, what an honour it is to be on the Smooth Brain Society podcast. Honestly, you talk about the Oscars, you talk about the Golden Globes, and you talk about the Smooth Brain Society podcast. It is truly an honour to be here. It's truly an honour. My origin story, I started off at university studying medicine, and I did a couple of years of medicine, and it was okay, found it interesting enough. But in my third year, what we had to do when I was at university at UCL is that we had to do our third year in a different department, doing the final year of another degree. And I ended up doing the final year of a medical physics degree. And it was a bit of a punt. I didn't. It was a bit of a. But it was a bit of a. It was a bit of an out of the box choice. I liked physics at school, and it was a bit of a toss up for me between physics and medicine. And it was really nice to sort of bring the two together. And I just really, what I really enjoyed was going from first principle, going from an equation to understanding how does this machine actually work. I really enjoyed that, and I really enjoyed diving into the theory and then coming out into the real world. As I towards the end of my third year at university, I became really interested in pursuing a PhD. Chased all my lecturers asking, "Do you have any positions available?" And I started my PhD a few years ago now, in 2017. Looking at how light travels through the baby brain and how we can account for the fact that babies' brains grow as they age. Is that really an answer to your question? But is that an origin story? That's just a really bit boring. That's just going in chronological order, isn't it? It's okay. We'll edit all this out. I think for me, what I really enjoyed is, I don't know, what do people say for the origin story?
Exactly what you've just said, like spot on. Like this, like, and that's a really fantastic story. Like I didn't know you'd went into medicine, and then actually for medicine, you're like, no, what's kind of the intersection between the two, like physics and
Yeah,
medicine, bioengineering
yeah,
and neuroimaging. Like that's
yeah.
yeah, exactly. That's like fascinating. So you, and I had no idea you went into medicine. That is wild. So you're basically a physicist and like a medical doctor
Well,
almost.
I might need to finish. I actually no, I do need to finish off this origin story. So I was doing my PhD, and I really enjoyed it. I really enjoyed sort of transitioning from seeing myself as a student to seeing myself as a scientist, and shaping the direction of my PhD, making decisions, thinking, oh, based on the data, this is the direction we should take. And it made me realize towards the end of my PhD, I want to go into research. I really enjoy science and the process of discovering new things. And so I did a bit of soul searching in the last year of my PhD, and I decided to pursue a career in research as opposed to going back in and finishing my medical degree. So I started off doing medicine, and I only did 2 years of it. And yeah, so I went to pursue a career in research. I finished my PhD, got some fellowship funding to do some work looking at baby brains, and basically developing a new method to look at baby brains. And then it was from there that I sort of got more interested in the more practical elements of science. My PhD was mainly sort of the computational stuff, and I just really enjoy sort of bringing together the sort of diving into the data, trying to work out what it's all about, alongside with how do we get something to work in the real world.
That is a really, really good origin story. Coming back to Beth's point though, it's quite funny that you enjoyed both physics and medicine. Most people I know pick medicine because they hated physics, or the vice versa. They hated bio, so they go into engineering chemistry.
Yeah.
You're one of the rare specimens.
Yeah, I think what I enjoyed was when I was studying medical physics is sort of thinking for myself and thinking, right, how do we work out how this thing actually works, rather than just memorizing things. And I did enjoy a lot of the things that I studied when I was doing medicine, but I just really enjoyed sort of working things out and understanding how they work, going from this simple equation right up to how do we measure oxygen changes in the brain. That's what I really enjoyed.
So, how do we measure oxygen changes in the brain then? Is it one of the things you actually use in your research day to day?
Yeah, so a lot of my work has been with a technique where you shine light into the brain, and it can map where there are changes in oxygen levels in the brain, which is a marker of brain activity. So that's a technique called near-infrared spectroscopy. Basically, we shine light into the head. This is red light and light that's just beyond the visible spectrum in the infrared range. And this light is quite good at traveling through tissue, fairly deep into tissue, maybe up to 2 centimeters. But the light, when it gets into tissue, it gets scrambled very quickly. It forgets its direction. You shine light into tissue and it's a clusterfuck. So what happens is some of that light that you shine into the body reflects back out. And we see this if we shine light into the head, it can travel through the skin, the skull, into the brain, and some of it reflects back towards the surface. So if we can measure how much light reflects back towards the surface, that can tell us about what's going on inside the brain. And that's particularly interesting because when a part of the brain is activated, more blood flows to that part of the brain, and blood is better at absorbing light than the surrounding brain tissue. So if we see a drop in levels of light that reflect back towards the scalp surface, we can say, there must be brain activity going on there. So that, in a nutshell, is how we use light to measure changes in oxygenation, which is a marker of brain activity.
Okay, so you've spoken about how your kind of imaging techniques work. And why was it specifically, I know you briefly touched on this before, you're looking at babies over adults in it, and is there any differences when you look at those two?
Yeah, I think a lot of what I did in my PhD was thinking about how babies' heads grow and how that affects the way that light travels through the baby head. Now, what got me interested in adults was thinking about times of life when we see changes in brain structure. And it's back in about 2021, 2022. I started collaborating with a group in the Department of Physics at Cambridge, who are interested in dementia. And what so the lab's PI got in touch, and the question she had was, how does the fact that we see atrophy in the brain, how does the shrinking brain, how does that affect how light travels through the brain? And that really got me thinking that it's the same thing that we're seeing in babies. Structures changing with age, but it's sort of, rather than things growing, we're seeing things shrinking. And that's what initially got me interested in dementia, was that technical angle. And then more and more sort of looking at the sort of impact it has on society, and seeing what the needs are, and also with my own story with my grandmother who got dementia, and seeing the real value in working in that space. So that's where the sort of transition from one end of life to the other came from.
I might ask quite a simple question here. Could you just sort of describe dementia a little bit? So, for people's understanding, usually when you think of dementia, you think of sort of memory loss. But uh, could you tell us what's actually happening in the brain? You said like it's shrinking, but what
Hmm.
does that sort of mean or look like?
Yeah. So, in terms of defining dementia, I'm not going to give a definitive explanation because I think there are experts out there better to answer that myself. But it's a change in the way of how we sort of perceive and understand the world, and what we see is shrinking in some areas of the brain, particularly that layer of the brain, the cortex. We see that actually start to lose quite substantial amount of brain cells there, and that's what can impact things like how we understand the world. And from my perspective, what's interesting about that is thinking how deep does light get into the brain, and if we're going to use the techniques that I've used before, the near-infrared spectroscopy techniques, how do we know that the differences in signals between one group and another, is that down to genuine changes in how the brain's functioning, or is it just down to the fact that the brain's deeper, and that the levels of light getting into the brain are lower? And that was sort of the interesting technical challenge that got me interested in dementia in the first place.
Oh, yeah, it's fascinating. So it's like it's from what you're saying, it was like the opposite problem to the baby's brains is there was a shrinking brain. So kind of going back because I feel like there was kind of two separate questions there. Was with the one where you were looking at the kind of the light in the baby's brain, was that literally just to see how it was growing, or was there a specific question that you were answering?
Yeah. So I wanted to do a PhD looking at near-infrared spectroscopy. So there was one of the lecturers when I was studying medical physics. Her research was in near-infrared spectroscopy, and she was heading something called the Brain Imaging for Global Health project, which was following two cohorts of babies, one in the UK and one in the Gambia. And the idea was to see what the effect of malnutrition and other early adversity was on baby brain development, and see if there's a difference between this UK cohort and the Gambian cohort. And what I found particularly interesting about that was we develop these techniques to image the brain in labs, but we don't think we only want to apply them in high resource settings. And what sort of really captured me about this project was that we're applying these engineering advances in low resource contexts, and thinking that there's so many people, there's such this populations that we can get data from and better understand. We can use technology to better understand how brain development is in different populations. And so when I was studying my PhD, there was the question that my sort of thesis centered on was, are the changes the sort of question my thesis centered on was, as the baby brain grows, how does that affect how light travels through the head? And can we account for that so that if we see changes in brain function, we can say those are changes in brain function rather than those changes being just down to changes in anatomy. So that was the interesting question when it came to babies, and that's what sort of was guiding my work. So I thought a lot about how the baby brain grows, what changes we see in the brain, and we see really quite substantial changes in the brain, particularly when we think of babies born, babies just as they're born, if they're born preterm or a term age, and how quickly the baby brains change.
That's fascinating. Um, so, so did you see any differences? Sorry, did you find any differences between sort of deprived conditions and I guess the UK? I'd argue, depending how deprived you think the UK is as well. Would you also see, if, to that regard, would you also see differences, suppose, say, within UK populations as well, based on indices of deprivation, where they are in the UK? Yeah, those kind of things.
I think those are really interesting questions, and I know there's been work since on data from the brain image in Global Health Project to try and answer some of those questions, or similar to what you've asked there. Um, my job there was more sort of on the technical angle, and that's what I really enjoyed about that project. It was crossing different boundaries. We had people working on the physics engineering problems, also people working on the psychology, the neuroscience problems, and others thinking, how do these findings translate to society? So for your exact question there, my answer is but there is work out there, and I'm pleased to have sort of helped contribute towards that.
So what does a physics problem in this look like in this sort of scenario? Do you need to sort of, I don't know, I guess like with the tech, do you need to like alter it or change it based on location who you're measuring? Yeah, so I'm just trying to figure out like your role and
Yeah,
the project.
yeah. I think so. What was interesting is I think one of my findings was when we look at babies at around sort of 6 months to a year, um, the differences in brain anatomy don't have too much of an effect on how light travels through the head. But what really matters is how accurate we are in placing the sources and sensors of light on the head. That has a big impact. And so part of my work was showing how we can increase the accuracy of baby brain imaging data using near-infrared spectroscopy, use how we can basically improve how specific we are and how confident we are in locating activity in the brain. So that was sort of one of the findings from my PhD.
And are you able to kind of look at specific areas in the brain? Like, I think one that we maybe briefly mentioned that people might know of is the hippocampus, so like the memory region, or is it more kind of like on the top of the skull rather than those top of the brain rather than those deep structures, or can you look at all of it?
Yeah, so when we're using infrared spectroscopy, it's the outer layer of the brain, the cortex, the bit that's just below the scalp. And in terms of regions, when it comes to say cognition, the regions that I'm particularly interested in are sort of in the frontal areas of the brain. An area I'm particularly interested in is something called the inferior frontal gyrus, and has functions in things like language um, and there are some other regions that I'm interested in when it comes to sort of thinking of the dementia context, is thinking of regions that are involved in processing sound, and those are sort of at the side of the head in the temporal regions of the brain, things like superior temporal gyrus, the temporoparietal junction. Um, in terms of something like the hippocampus, that is really important, particularly when it comes to memory. Um, Unfortunately, that's just too deep to get light towards. But it's thinking, right, what things connect to the hippocampus, or what sort of in that network of brain regions, what are the things that we could measure that are on the surface that we could use then to infer about functions that sort of involve the hippocampus.
Oh, makes sense. It's like constant problem solving almost. Can't get to that area, but what can we pull? What can we from that from other areas?
Yeah, and I think it's understanding that sort of different brain imaging techniques have different Achilles heels. For instance, MRI is fantastic, and that will give you a high resolution look at the entire brain, but it might not be particularly useful or scalable in different populations, and that's where we sort of bring in different techniques. Is all of the different traits and the trade-offs, and I think that's something that's really interesting about sort of innovation in the sort of medical imaging space.
Yeah, oh, the other one I was thinking about was the cerebellum because I guess it's reachable, right? It's pretty important in sort of coordination and things like that. I was just thinking about babies and motor coordination. I was thinking that's a cool region to look at or study.
Yeah, cerebellum is really, yeah, it's really, yeah, I think it's a really interesting region to look at. I think I've not sort of attempted myself to measure it. I think in terms of its depth, it's fairly, it's not deep, but it's deeper than a lot of bits of the cortex. And I think in terms of the specificity you need to be able to study the cerebellum. I think something like near-infrared spectroscopy in its resolution might be just a bit too coarse to be able to give sort of the more fine-grained sort of spatial data that you need to make inferences about the cerebellum. But what I like about the cerebellum is, so whenever I'm doing comedy, sometimes I want to take a temperature check of what the audience is like. Do we have a lot of geeks in the house, or do we not? And so, I mean, I've 3D printed my brain before, and I say, I've 3D printed my brain. I call my brain Cera, short for cerebellum. And in fact, I always keep my brain handy. But yeah, this is
your actual brain.
This is cerebellum. Yes. But yeah, you see some people holding models of the brain, and I think, have you actually got your brain 3D printed? I was talking with someone a few months ago, a lecturer at UCL on Zoom, and we're just about to finish the call, and I said, "Is that your 3D printed brain behind you?" And he said, "Yeah, it is." And he was brought it to the camera, looking really proud. And I said, "I've got my brain 3D printed as well." So it's a flex. It's a flex.
It's a definite flex, and yeah, especially like, do you say you have got the cerebellum on it as well?
I've got, yeah. It's not particularly. I probably could have done a better job on this, to be honest. But yeah, there's because I remember from doing being in the anatomy lab looking at brains when I was sort of in my undergraduate. Yeah, the little like layers of the cerebellum and just how, yeah, I remember. I haven't quite got that level of detail here.
I mean, it would be hard to. I mean, there's a little fact for everybody. There's more neurons in the cerebellum than there are in the cortex. Understudied, I would say. There's lots going on there, but so it's an interesting part of the brain, or sometimes called the brain.
Another fact: there are actually more cells in your brain than there are atoms in the universe.
That is a wild fact.
That cannot be true.
You pull in a face like,
it's not true. That's bullshit.
It's very much. I mean, it's very much. It's the.
You had a little stick on.
It's the end of Amélie, is it? It's the French film Amélie. Is it at the end, and the narrator says about somebody finding out there are more brain cells than atoms in the universe. I remember thinking, that is amazing, but
how accurate is that? I mean, it sounds cool.
That's not at all accurate, because like each brain cell requires so many atoms to make. Like it falls apart immediately, Liam. That statement.
You could say there are probably more possible connections between brain cells than there are atoms in the universe. That I don't know. That might be true. I don't know. That's more like that. That's. I mean, then that would be 90 billion factorial, which I think is a very big number. I've never calculated it myself, but is this going to make the cut into the podcast?
This is making the cut.
All of it.
I quite like how wrinkly your brain is as well. And coming on the Smooth Brain podcast, it shows the difference between a smart person and us.
I mean, took maybe my brain out, wouldn't be quite as wrinkly as that.
I mean, it's definitely not.
Yeah, I quite like this. I'm quite proud. I'm quite proud of my brain. It's the other thing about this brain is when I printed this brain when I was in like my fairly early on in my PhD, and it was at that point where I remember after I printed this, I was like, oh god, I don't know how to tell left and right in an MRI scan, and basically this brain might be the wrong way around. So the right might be the left, and the left might be the right. So this is either my brain or it is a mirror image of my brain.
Selfie of your brain.
So it's a selfie. Yeah, it's a selfie.
Okay. Would that mean that you were...
Oh my god! I'm making sound. I'm going to put that. I'm going to touch it. I'm making too much sound. I can't touch the table.
Does that mean if you are right-handed, then if your brain was flipped and you put that back in, would you become left-handed? What differences would there be?
Oh, you've nailed neuroscience there, Beth. You've nailed it. It's interesting. I think there's the right ear advantage you have, which is where it's easier, or people tend to find it easier to perceive speech with their right ear than with their left, which I believe is down to sort of how the wiring in the brain rather than the actual structure of the cochlea of the ear. So you might, if you put it in the other way around, you might find that your left ear is more tuned in speech.
And
Not
the other way
particularly
around,
scientific, but
considering the left hemisphere of the brain is more used in language, we swap the brain sides around, then your right hemisphere will be more used in language.
yeah, I think it would be. I think
Theoretically.
yeah. I think it's an interesting thought experiment.
Yeah. I'm going to quickly go back to, because I've this little gone off a little tangent, which I love.
That might be my
Because I've got a couple more questions about the wearable brain. Because is it you can wear them? Because I think most of the time that maybe what people don't realize, when you have your brain scanned, you go into like a massive machine. You know, it's solid, it doesn't move. But with this like wearable brain imaging, how close were you using that to in clinics? Sorry, a couple of questions. Close to using that in clinics. You commented that there was a study between like women looking at health, brain health in Gambia and England because we use them in Gambia. Like, what kind of like uses does that have, especially I guess with in your research as well?
Yeah, I think it's. I think what I'd like to see these things sort of used in clinic because I do believe that they address a need that we have to get brain specific biomarkers. So a lot of these devices have mainly been used in academic research, but have also been used in clinical context as well. One of my colleagues actually last week was covered in the BBC. Flora 4, who's been applying these techniques, these near-infrared spectroscopy techniques in babies. Some really really interesting work there. I would in terms of how it's actually been used, it's mainly it's mainly been in academic research. Now, in terms of what it is actually like, so imagine you put a cap on your head, and as you were saying, Beth, usually when we think about going for a scan, you think of lying down flat in a big machine that could be loud. But here I'm thinking, we're sort of thinking of this differently, and we're thinking, right, put a cap on your head that's made out, so think of a swimming cap. So the caps that I use are made out of basically wetsuit material. So think putting wetsuit material on your head, and in that you've got sort of little hexagony things. In fact, and think about in your sort of wetsuit cap, you've got loads of little sort of hexagony things like this, and you've got that dotted all over your brain, well, all over your scalp. And the idea is that we don't need people to go into a big bulky scanner. We could then have something that could see what the brain is doing in different environments. We could take it to a point of care setting. It could sit in a drawer to GP's office, and could be useful to get brain biomarkers from someone in a sort of more convenient location, rather than having them to go off to a specialist centre.
I think my immediate follow up to that is something like if you're putting a cap on, the other thing which I think of is sort of the EEG caps and things, which I guess this is kind of similar, but they often have a problem with people with hair. So people with thicker hair generally it's a lot harder to collect data from them. Does this also suffer from similar problems? I feel like thicker hair would mean that light going through your scalp would be a lot harder. Do you come across some similar issues?
Yes, hair can be a problem. Yes, and
Scalp density as well, I guess.
yeah, I think here's a problem. Basically, what you want is you want the underside of the cap to be in really good contact with the scalp. And this is something that I've found in the lab recently, that sort of participants that I have in the lab, and I'm scanning them with these caps, if they've got no hair, great, we get great signal. But to get people with, when people with hair come into the lab, I need to do quite a lot of work to sort of almost massage the cap so that it gets into better contact with their head. So we can think of like the other side, the underside of the cap. It's basically got lots of sort of, let's say, 3mm long tubes, about sort of 3mm odd in diameter. So it's like there's lots of little things poking out that I'm trying to massage through the hair, sort of comb it through the hair. And so some people in the lab, I spend quite a bit of time, particularly with the thick hair, getting it through. I think hair used to be more of a problem. It's getting to be less and less of a problem. But it's still something that I've been putting a lot of work into to sort of try and minimise the effects of hair. Unfortunately, I can't just shave people's heads when they come into the lab. I make the joke with every participant I have, and people find that funny. It's interesting when you get a lot of people into the lab, you just say the same jokes again and again and again, and you're like, wow, that is funny. That's the millionth time I've heard that, but they still find it funny. But yeah, hair's an issue, but I wouldn't shave my head for some.
Well, I guess it was coming to the question of, you said, like, going back to thinking about the study about Gambia versus the UK. I know they're babies, but once they start growing, the hair also was different. And if the study was a few years ago when you said it was more of a problem, now the same readings can also be interrupted or have more noise to them, if you know what I mean, than what would otherwise.
And I guess maybe this is why you're looking at babies and people maybe towards the end of their life, because we kind of don't have hair at the start and we kind of lose hair later on. I see the reason you've chosen these populations, Lee.
Yeah, I think
All makes sense.
babies are great. Yeah, great signal with babies. But yeah, I think the hair's gonna become less and less a problem
It's going to have a whole section here on hair.
it takes up a lot of my it's taken up a lot of my thinking recently. How on earth do I deal with hair? I joke that hair is the enemy to my participants, but I'm not joking. But we are solving that problem. We're solving that problem of how to deal with hair.
They get a free head massage as well. I hope that you put that on your posters for when you're recruiting.
I didn't put that in the sort of ethics application that participants also get a head massage. So I'm not sure I could advertise that. However, I do often get sort of, as I'm trying to massage the cap onto people's head, they say, "Oh, you could be a hairdresser," and I make the joke that if science doesn't work out, could I use this as experience on my CV? And apparently people find that funny, so I keep saying it.
Yeah, I find it.
Yeah.
So,
sorry, go ahead, Beth.
No, it's okay. It's kind of moving on to a different point. So you.
No, there's just one more thing was around. Do you use gel or anything to put the cap on, or is it just like those light tubes sending light straight down? I'm just thinking similar to EEGs, where you have like water or gel which you use to stick the caps.
So no gel. So I get participants coming to the lab and they're saying, "Oh wait, I don't have to put gel on my head," and I'm like, "No gel today." As I understand it with the EEG, you need to match the impedance between, or have some intermediate in sort of electrical impedance between the scalp and the electrode. But with this, no, I think. But the key thing is to make sure there's good contact with the head. So at the end of experiments, the participants, I take the cap off their head, and they've got lots of little sort of dimples, particularly on their forehead. Basically like wearing a tight cap. I like to see that because that means that they've been wearing wearing the cap correctly, and we're getting a lot of light into the head. So there's the challenges of trying to get light into the head, but we don't need to put use any gel or any of the messy stuff.
And so you use cap in babies, use cap in people who have possibly have dementia. With your new fellowship, what is that letting you? So I think it's the Blue Sky Fellowship. What does that let you do, which you couldn't do before? What's particularly great about the work that you're doing now?
Yeah, I think so. I started back in May this year on the Cambridge NeuroWorks Blue Sky Fellowship, and I think the first thing that came to my head when you said that was being supported to work at a quicker pace. And that's something that I've really enjoyed, and that I've sort of felt supported by the NeuroWorks team to work at a quicker pace and think, I've got an idea, let's test it out in the lab, sort of what needs to be true for this to be sort of implemented in future in clinic. And if we test it out and that idea doesn't work, that's great. We can then sort of pivot and think about something else. So it's been support to work at a quicker pace. It's been introduced to ways to ideate. So the first 3 months of this fellowship, I was given 3 months to basically come up with an idea to consult sort of the ecosystem when it comes to when it comes to neurotechnology, when it comes to sort of academia, startups, industry, clinicians, and think, what is the problem at the moment? What am I trying to achieve? And that was where I sort of pivoted from envisaging that this would be a hardware development problem that I'd be working on to thinking, no, I think this is more of a software development, more of a data analysis problem that I'm trying to solve. So the work I'm doing at the moment is I'm interested in dementia, and I'm interested to see how we can use emerging technologies like near-infrared spectroscopy to streamline the diagnosis process and offer clinicians a brain-specific biomarker that could be used to improve diagnosis times and improve clinical certainty in diagnosis when it comes to thinking of dementia subtype. The idea I'm exploring at the moment is how listening to things, or how cognitive. The idea I'm interested in at the moment is how listening and cognitive tasks that rely on listening, can that be used to offer clinicians a brain-specific biomarker that could be helpful in dementia diagnosis. What I'm doing right now is piloting an imaging protocol for this in healthy participants, and my hope here is to see what sort of conditions in that experiment elicit the most reliable brain responses. What are the parts of the brain where we see robust responses that could be useful targets when it comes to thinking about clinical diagnosis in a clinical population, and I'm hoping to apply that in future in a population with dementia.
Right. So would that be looking at, because I know you spoke about the kind of sound and the superior temporal kind of regions. Would that be something that you'd be looking at with sound, or is it more like processing the sounds?
Yeah, it's interesting. So it's interesting because sort of regions that are linked with auditory cognition as well as low-level auditory perception are based in the temporal lobe. So I'm interested in what's going on in the temporal lobe, also the connections to other parts of the brain, particularly frontal regions that are sort of involved in more general cognitive processing. And a region I'm interested in is the inferior frontal gyrus that we know is linked to things like listening effort and higher cognitive demand during listening tasks, as well as other more general cognitive tasks. So yeah, thinking about sort of the front of the brain and the side of the brain, those are the sort of bits that I'm most interested in for this work.
So, take us through, if I'm a participant in your pilot study, you put the cap on me. What happens then? Are you like playing me sounds? Are you doing a comedy bit which I listen to and you see the activity? What sort of, what kind of task does it look like? Are we just seeing just the base range?
Yeah, well, well, if I'm going to walk you through, the first thing I do is I'd meet you in the reception of the building. I used to talk about some pleasantries. Where have you come from today? What are you doing? Blah blah blah. Get you into the lab, talk you through the experiment. But no, in the actual experiment itself, people are my participants, my wonderful participants, are wearing the cap, and the cap's sampling from the sort of temporal and frontal regions, and they've got earphones in. So what they do is they squash down the foam in their earphones, put it in their ear, and then they sit looking at a screen in the dark for about 25 minutes. And what they're doing is they're listening to different things, and then I'm asking them to repeat what they've heard. And what I'm interested to see is sort of the responses to when they hear the sound, the responses just before they respond, the responses after they respond, to see sort of these different things that are happening from perception to cognition. And interested to see what the responses are in those temporal and frontal regions.
Nice. And are you expecting for possibly those? I mean, of course, you're still in like the testing phases with the controls. Do you expect that are you still looking at the blood? The blood flow, I guess. Would you expect there to be less blood flow for those who maybe are looking for a diagnosis of dementia or have dementia, or are you still unsure? Any little snippets of hypotheses you can give us?
Yeah, it's interesting because what I'm interested in right now is sort of seeing what regions. So firstly, to pilot this protocol um, ahead of hopefully then in future applying it in a clinical population. But I'm also interested to see what brain regions, what responses do we see in the brain, and then sort of focus in on those in a future study in people with dementia. So what I'm interested in. What was your question again, Beth?
Would you expect there to be a lower, lower
Yeah,
blood
yeah,
flow?
right. So in terms of what I'd expect to see in people with dementia or specifically people with Alzheimer's disease, um, I'm interested to see what the response is in this region and whether we might see an increased response, we might see a decreased response, and I'm not sort of committing right now to a particular hypothesis on this because we may see compensatory effects in the brain. So what might happen is we might see a deficit in one part of the sort of cognitive network, and then another part of the cognitive network sort of makes up for that. So um, we may see in someone with cognitive impairment an increase in activity in one of these regions. Um, so that's something that I haven't got a specific hypothesis right now, but what I'm really interested to see is what are the regions where we can get these reliable responses in healthy volunteers, and that is then a sort of that's then a sort of proof of concept of the neuroscience that these regions can be measured using this method. So let's go and see how it could be used in people with dementia.
That is so cool. So you're literally, it's really cool to see it's very quick. I mean, I'm running my own study and it's taken me 4 years to get ethics and finish it. It's wild. But this is just, you're like, yep, I have an idea. I want to look at where blood flow when we're having listening effort, which we spoke about Kate with a little bit, which parts of the brain are linked to listening effort and with blood flow. And then once you've kind of got those down, you can be like, oh, is this different in people with Alzheimer's disease? And maybe in the long run, can you identify different types of dementia? But a talk for another day, probably.
Yeah, I think it's interesting. I think right now I'm sort of in a proof of concept phase, so it's thinking let's show that this thing works, and sort of get pilot data ahead of a future study, and then sort of the next step in future would be maybe a bit more rigorous when it comes to thinking of sort of establishing a baseline and sort of comparing a control cohort to a cohort with Alzheimer's disease. But yeah, right now it's in the lab. Let's show this thing works. And yeah, that's something I'm really enjoying.
Yeah. The few One thing which I was wondering, because you said that the cortex. Oh no, well, this is usually for the cortex. You can't go much deeper, but then I guess certain dementias, start from the hippocampus or start from the medial or central regions of the brain. Those I guess would be a lot harder to detect from this. But I've now that I'm saying it, I'm thinking. I guess if somebody shows with symptoms of dementia and you put the cap on and you look, but sort of the lateral, the outer regions are fine, you can kind of then narrow it down by elimination that it's probably one of the more medial regions which are degenerating.
I
So
think
yeah.
what's interesting about that is thinking, right, as you say, sort of saying Alzheimer's disease, and we're seeing sort of the first sort of signs of atrophy and sort of medial temporal. Sorry, yeah, the medial temporal gyrus. Yeah, we wouldn't be able to image that, but it's thinking, what about other parts of the brain, and do we see changes in brain function before we see those structural changes? And it's thinking, right, okay, let's think about the network. Let's think about how the brain is set up. What parts of the brain are in contact, have strong connections to the medial, the medial temporal lobe, and thinking, right, which of those are on the cortex and which of those can we measure? So it's taking that sort of wider approach. We may not be able to look at the seed region, but we can look at somewhere that is very closely connected and sort of infer what's going on.
Fascinating.
Yeah, that's really interesting, and I'm excited to see where it goes. It sounds like it's still kind of it's a proof of concept, early stages. I usually ask a question here, but I feel like you're literally you're literally already doing it, Liam. I want us to pretend that you've been given a grant of £1 billion more, unlimited, and the ethics committee were on holiday and have said, oh, well, we trust you, you just do whatever you want to do, what would be your absolute dream study?
See, that's such a big question, because that's basically like you always think about research ideas in amongst these constraints, and usually amongst the constraint of a grant call. And now, with your question, it's like
Anything.
What I think what would be really interesting is to get people and scan them with different things. Let's scan them with EEG. Let's scan them with high density EEG. Let's scan them with MEG. Let's scan them with MRI. Let's do all these different MRI things. Get near infrared spectroscopy. Get different kinds of near infrared spectroscopy, and think what are get all of this data, get it from loads of people, and then think, right, what are the things that I would usually only be able to get with a big bulky scanner? What can I get using a much easier to use and cheaper device? And I would be keen to get, loads of data, and then we could see, it could even be something very obscure, sort of the link between this part of the brain and the signal and this frequency band in EEG. I think it would be really interesting to do that, because then we could sort of distill sort of these technologies and these really sort of advanced technologies like MRI, and think, use this cheap thing to get information that's just as clinically valuable. And that would mean that we could roll it out across sort of a much wider area, and then get more people to get the information that can sort of support their health and sort of their healthcare outcomes. I think that would be, I would scan a lot of people at a lot of different ages, with different conditions, with a lot of different techniques.
I feel technically that one's pretty ethical. It's just the money constraint on that one.
I've got a billion. I've got a billion in the bank here, so I'm it's I haven't done a power calculation on that, but yeah, I think it would be. I think that would be interesting.
Yeah, a bit like I know the biobank has lots of different bits of data, but biobank is like a massive, for those listening, biobank is like a massive database of thousands of people with lots of different measures, like age and some imaging and some kind of cognitive things, lots of different kind of metrics about people. But like this would be like the kind of the absolute gold standard imaging one, and yeah, it'd have every type of imaging possible.
Yeah. That biobank on steroids.
Biobank on imaging steroids. Yeah, like it. That we'll call it that as well. I think the ethics would particularly like that if they were around.
I mean, biobank does have blood biomarkers already, right? Because it does like blood tests and stuff. So you could, in theory, map them to your brain imaging.
Yeah, I mean, imagine if we could get something from blood biomarkers, that could tell us what we get from brain imaging. Now, part of why I'm interested in looking at at sort of getting a brain specific biomarker in dementia is to sort of complement the advances we're seeing at the moment in blood biomarkers. So blood biomarkers can sort of tell you things about what's going on in the brain, but I'm interested to get a device alongside that can sort of look at the brain specifically.
Yeah, well, I think it's important for people to remember, if you don't know, that there is a thing called the blood-brain barrier, and the blood biomarkers can tell you a lot about the brain, but they aren't directly the brain, and therefore it's quite important.
The BBB, as I wrote it in my notes, as shorthand.
Yeah, great. So I think, is there anything else you want to ask to hear before we move on to maybe the public engagement?
And the less serious stuff. Good. Oh, thank God.
But is there anything, Liam, you wanted to talk about which we didn't actually cover in terms of your work before we go on to the fun part?
Not really. To be honest, no. I'm quite happy with sort of how the conversation's gone. That it's sort of focused more on the sort of technically angles rather than because something I enjoy doing is trying to feel my way and sort of see how this works in the sort of diagnostic pathway. But it's not, but sort of that and symptoms of dementia, things like that. I'm just pleased that it's being sort of more technically guided, this conversation. So that would be the only thing that I would wanted to have said at the start. And yeah,
we can get someone more clinically focused to get into the nitty-gritty of the clinical aspects of dementia. You have to remember, people are very, very specialized the higher up you go.
Yes,
It's not
yes.
as simple as you think. Oh, I researched dementia. What part? Oh,
What
the
part?
techniques.
So I think I said at the start, you're part of Three Queers and a Welshman. Can you tell us a little bit more about that? How did that start? What kind of comedy do you do? I guess the origin story of that, maybe.
The origin story. So I think it's back in 2023. We started on a program called Talent Factory, and we met basically a science communication sort of program. And so I met Eva, and Max, and sort of the thing we had in common is that we liked silliness, we liked science, although we were in different parts of science, of course. But I think we really like science. We really like talking about science. Um, and so it was one day in the group. It was in the group chat. We saw that there was the Green Man Festival were put a call out for science exhibitions. And sort of I can't remember if someone put in the group chat and said, anyone fancy doing this? And the four of us were like, let's do it. We didn't know what we were going to do, but we sort of thought, right, what is it? How can we sort of bring our science together? And sort of we came up with this idea of a maths and neuroscience crossover. So we thought, right, let's do this exhibition where we think about maths and we think about puzzle solving, and then let's bring along a brain scanner and scan people's brains. And anyway, that was what we took then to Green Man in 2024. And then and so that was sort of the sort of public engagement bit. And then what happened was while we were at Green Man Festival, there was an act that dropped out on one of the stages. And so we were asked because we knew the person was running the stage, and she was saying, could you fill in an hour slot? And we were like, yes. So we filled in with an hour long show on science comedy, and yeah, in terms of the name, basically how this came up was we were putting a proposal together for Green Man. It was a question on quality, diversity, and inclusion, and we thought, right, what can we put for this? And we wrote, we are three members of the LGBT community, and there is also one Welsh person, and that's it. I can't remember who came up with that name, but we thought, let's just perform under that name, and so that's what we did at Green Man Festival. We thought, what, we could, as well as the public engagement staff, why don't we give why don't we sort of try and bring this together for a science comedy thing? Because we met, the four of us met doing science comedy, and yeah, that was sort of the origin story. And then when we went to Glastonbury, we got asked to do to do a few performances on a stage there. And yeah, we were like, let's do it. So yeah, it's really really pleased how that came together, and really pleased to have three other people who are just as committed to silliness, nonsense, and science as I am.
So wait, did you just name drop Glastonbury there?
I did just name drop Glastonbury. So we did three shows at Glastonbury. So we the one of which we got a spot on Robin Ince, the who hosts Infinite Monkey Cage alongside Brian Cox. He had a show called 9 Lessons for the Summer Solstice. And so we were asked to be on the bill for that, and we were so pleased about that. So we had a 10-minute slot for that show. And we thought, right, what we're going to do is we're going to pack as much comedy as we can, much comedy and science into 10 minutes as we can. And we came up with this fantastic set where we all came together to talk about consciousness and what is consciousness, solving the puzzle of consciousness. And I was really pleased with that. Robin introducing us as three queers and a Welshman was fantastic. And yeah, we did a couple of other shows at Glastonbury as well, sort of as sort of more. We sort of fleshed that out a bit more, sort of our nature of sort of solving puzzles, but making it funny at the same time, and bringing in some science. And yeah, I think bringing science into comedy is great. I feel like in comedy you are invested in someone's story, and part of the storytelling aspect that there's something that I like, sort of someone immersing you in their world, and yeah, why not do that with science? So yeah, so that was I did, to answer your question. Yeah, I did name drop Gus.
That is like honestly like the coolest thing. I remember, yeah, when you first said that, that is wild. So I got a lot of the audience, I guess you see, aren't very science based. How do you find different audiences react when you kind of mix the sciencey maths and the kind of the humour together?
Yeah, so it depends. Some audiences are performed to much more sciencey than others. That's where the cerebellum joke comes in.
Yeah,
Try and see, right, what audience am I with today? And I think when it comes to science comedy, sometimes you have to make a judgment call. Am I going to go sciencey or am I going to go funny? Would something funny be better here or something sciencey? And most of the time, funny wins. And I think that to an audience that's not immersed in science, anything scientific is really new to them, and sort of as scientists we can sort of forget like that this isn't the usual experience. Being surrounded by science all the time isn't usual experience. So I think a lot of the time is thinking, how do I make this funny rather than how do I make this more sciencey? But that's still a great way of bringing in science and bringing it into to audiences that aren't scientists. And yeah, I do really enjoy talking to non-scientists because they bring a different perspective, they bring different ideas, and that's I just really enjoy talking about science, and I just like being silly about it as well, and saying silly things about it on stage.
Yeah, makes it fun, doesn't it? And so how did you, I guess kind of two questions here. How did you get into it? And I know because I've kind of been dipping my toes in the past year or two, and I know the people who are similar. How do you, because you're quite established now. So first of all, how did you get into it? And what advice would you give people who want to get into it a little bit more?
I mean, Beth, you say dip your toe in it. I'm talking to the person who has conceived Amy Godala.
Oh, I know. Amy if I brought Amy here right now, she would she would be saying she was the expert. Unfortunately, it's just Beth today. We'll keep it modest.
But I mean, Amy Godala is also a bit like Cerebellum in the sense that you
Yeah.
need a level of knowledge about the science of the brain to get the statement. Amygdala.
To answer your question, so I mean, my advice to people would be just get involved and look for a sort of a sort of science comedy community, and it's around the place. You've got things like Bright Club that are at different different sort of cities around the UK. Dip your toe in that. If you know someone who does it, send them a message about it. I had someone that got in touch with me about getting into science comedy, and then we bumped into each other at this conference, and I was like, oh wait, we know each other through silliness, not through the serious science that we're talking about now. So how I sort of started out, so I did some gigs in sort of different venues in London, and that's how I got to know the other three in Three Queers and a Welshman. And I think it's terrifying. It's absolutely terrifying, but you learn a lot from doing it. And when I was an undergraduate at university, I did stand up for a couple of years. And I think one thing that I didn't like about that was I wasn't able to talk about what I wanted to talk about. Sort of science comedy, sort of finding a way and sort of finding different audience that are interested in science, might not be scientists themselves, but interested in science. I think that sort of helped me sort of find a voice in this space. Now, my advice would just be you're going to need to feel terrified, because it is terrifying, and it doesn't get any easier. I'm not a I'm not a it doesn't get any easier, and I think and I think sort of you sort of have to try new things a lot, and I feel like every set that I do, I try and do something new, because otherwise you don't know what's going to work, and sometimes you really don't know what's going to work. You can't just work it out when you're doing things on pen and paper. And I think I find that sometimes you write something down, you're like, that's it, that's going to be brilliant, and then it falls flat, and then you come up with some off-key remark, and you're like, wait, I'm going to sort of build around that in future. So. But yeah, but I think sort of thinking what's funny is sort of the way to frame it, rather than thinking, I want to talk about this particular science thing, is thinking, that's a funny thing in science. Let's talk about that. So I'd always try and sort of lead with what's funny.
Great advice. Well, the different sets thing is quite interesting because I've only seen one of your performances, and you were making the setup almost on the day, like 10 minutes before the show. You were still like, "Oh, I'm gonna add this. I'm gonna add that." You then coming back saying that it's terrifying. How are you so flexible to just prepare and say something 10 minutes before, but also be completely terrified about doing it?
It reminds me of my first gig when I was in my second year at university. As in UCL Comedy Club, and they were doing their freshers gig at the start of term, and I turned up to the taster session. They're like, "You're going to do the freshers gig, aren't you?" And I was just like, "Yes." I really didn't want to say yes, but I was just like, okay. I didn't feel I could say no. I remember being so nervous for that. So I've had a 5-minute spot. They're really nervous. And then I just remember sort of getting in front of people and thinking, I'm so terrified, but like, it doesn't matter. It doesn't matter. And you sort of become, I wouldn't quite say a different person, but you just become someone different. You sort of have a different outlook to things. You've got your stage persona. And for me, I find that that's a lot more confident than I usually am. Even though you're bricking it inside, you also feel like it's an odd thing. And I remember sort of in that gig, I mean, it wasn't particularly funny, but there was like, I was like, why am I going on? Why am I saying this off-key remark that's then getting doing this, saying this offhand remark that's actually getting a laugh? Like, what am I like? Wow, okay, so that, and then I've just found that afterwards, I write things and then I just rip it when I go on stage. I'm beforehand, I'm like, right, I need to sort of orchestrate this really well. I need to choreograph this really well. And then you get on stage and you're like, no, I don't know what it is. I'm just like, no. And so I've tried to lean into that a bit more and think, you know what, like, I don't need to be so prepared. Or I can sort of prepare, but then leave sections where I'm like, you know what, I'm just going to go with whatever's going on. I remember once I made a funny joke about the Fibonacci sequence because the person before me just mentioned Fibonacci sequence, and I was like, how on earth did I get such a big laugh to a joke about the Fibonacci sequence? But yeah, it's I don't really know that much about the Fibonacci sequence either. But yeah, I think it's just like, yeah, I think it's all part of the fun. It's all part of the fear is part of the fun. Part of that fear is I actually don't know what's going to happen.
I guess it's almost leaving it as islands. You've kind of got a little islands of funniness, but you have not like scripted it into like an inch of its life, which I guess is probably a good thing.
Yeah, and just but you also just don't know what's going to be funny. So it's almost like it's like the thing of, don't bother worrying about things, because what you need to worry about, you don't even know right now. Don't worry about it being bad on stage, because it's probably going to be bad anyway. So you may as well use it to try and sort of gather data on what's funny and what's not.
Yeah, that's a good way that's a really good way of it. Gathering data and what's funny and what's not.
Failure is a data point there.
Failure is a data point. Gathering data on what's funny and what's not.
Bet's writing that down for advice.
I am.
It's going to be on like a throw pillow sewn in now.
It's going to be on your wellness podcast. Failure is a data point.
Amy has taken that point and has now said it's hers. She said she fed that to you. She's just told me.
No, thank you. Thank you, Dr.
I guess fun little question is if you could get any scientist, dead or alive, to do a stand-up set about their research, who would you choose and why?
That's actually, that is interesting. Who would I get? I mean, you know what? Yeah, I mean,
Hair
he'd just get away with that. What he'd do is he'd mess up the hit, wouldn't he? He'd mess up the hair even more, and then he'd just get a laugh. Just like, he'd
being him,
basically do a
yeah.
Milton Jones impression, and he'd just get loads of laughs. I can imagine him sort of with sarcastic one-liners, you know. Um, who would I get back dead or alive? I mean, I think what would be interesting, and maybe David Attenborough. So, maybe not the conventional definition of what a scientist is, but I could just, if you had David Attenborough, this national treasure, or if you gave him a bit of an edge, and what if, you had him really wholesome, and then he just said something that was really, really off brand and really cutting, and sort of, because you imagine him as, he's the nice person, encouraging. What if he was just sat? What if he was just really, what if he was just sort of saying a sort of, making a few dad jokes, and then something really cutting about something in the front row? And I can imagine sort of in his accent, and it would just be so out of character. And then talks about some really odd science to do with, I don't know, how pelicans. Fill that in with your own imaginations. Yeah, yeah. I think that would be quite funny.
Yeah, David Attenborough is a with a character. I have
I
to give
think
him a name.
yeah,
I was just thinking David Attenborough describing a researcher like he would like an animal.
yeah.
So it's on stage.
The natural
Here we
habitat.
have a PhD student.
You could imagine that, and sort of commenting, yeah, that you've got a big conference deadline coming up, and everyone's in the office, or everyone's really stressed, and sort of commentating on a stressed conversation between two PhD students. I could get behind that. I think the issue is, I think you'd want someone who was a bit more understated. You wouldn't want someone with sort of who was a sort of a good scientist and they know it. You'd want someone a bit more. I think you'd want someone a bit more understated, a bit more humble. That's why I'd go at Attenborough. I think it's a really good question. But that's what you say, isn't it? When people say, "Oh, that's a really good question," I'm like, I know it's a really good question. I asked the question.
It may have been like, Beth, there's not that many. Like, I do not care for, some of the scientists, or maybe they just wouldn't be. But very funny. I was trying to think of some good ones. I was thinking of maybe of some like old charlatans in the past, like when X-rays first came out and somebody X-rayed the brain and it was actually pig intestines. I'd love to have the lead researcher on that.
Was that a thing?
Yeah, I've got an image of it in one of the lectures I teach. I'll send it over. It was amusing.
That's incredible.
I'll send you the image as well. I'll send you the image. It's funny. But I think David Attenborough, yeah, in character, great.
Should we ask Liam about his hot take now? While we're at
Yes.
it, while we're asking him questions.
Yes. Okay. Are you ready? So Liam, what is your hot take?
My hot take is that scientists don't know what they're doing, and that's in fact a good thing.
Oh, go on.
Well,
Could you explain on this?
Yeah, I think that as scientists, we're sort of the incentives and sort of the model is, and the funding is it feels as though we've got to show that we know exactly what we're doing, when in fact we should be doing science. We should be discovering new things. And part of that is not knowing what you're doing. I don't know exactly what I'm doing. I have a hunch. I've got an idea. I'm not completely shooting in the dark. I have some idea, but I'm trying to discover something new. And I'm taking a bit of a risk here. This might be me going down a rabbit hole and not coming back with something useful. But I think that for me it's scientists should have no idea what they're doing, and that's a good thing. And when we think of sort of big discoveries, it's not, a lot of things haven't just come from sort of incremental incremental improvements in how we understand things. And I feel like the current environment where we need to sort of show that our research is particularly safe, or it's going to say what we think it's going to say. I think we could have better and sort of more interesting scientific discoveries if we sort of let scientists do what they do best, which is discovering new things and learning how to do things when they're not 100% sure how best to do things. So that would be my view on it.
I think that's a very good point. I think, yeah, the system itself kind of like also limits what you can do because you're trying to prove that you know exactly which is going to happen and what you're going to do. But it also then stifles sort of innovation and things like that. Because if you could say, I don't know, but I'm going to try this, it's not like a complete shot in the dark, but there's some logic to it. Let me try it. You're probably going to get more innovative sort of ideas or findings.
Yeah, and things will fall flat on their face. Things will fall flat on their face. But there'll also be things that don't, and I think, I feel like, when I've been writing grant applications and sort of having to show, basically to say, I'm confident that we will get this result. I'm thinking, but I'm not, or I'm thinking, I feel like the real spirit of science is, I think this is going to happen. This is my prediction, but I'm not 100% sure. Let's give it a go. Let's work out how to do this. And that's my thought, that I think, yeah. And I think us as scientists, we're really good at that. And I think we should be able to do more of that, to do more of having a hunch, not being 100% confident about it, but giving it a go.
Yeah.
I have another hot take.
Go on.
Go
on. Why not?
Well, there's the one that I WhatsApped you. So my other hot take is that human beings are just the same thing as pints of craft beer. And when you shine light into the body, what you get, if I shine light into my finger, you'd see that my finger would glow red. Now, what's happening is the red light is getting into my finger from say, a white light source, like on my phone. But what it's doing is it's getting through my finger, it's getting through the bones, getting through the skin. So it's not being absorbed, but it's being scattered. So I can't see the beam going through my finger, but I can see my finger glowing red. The light's coming out my finger from all these different directions. The next time you have a pint of cloudy beer, say you're on one of your pretentious nights out drinking beers that just don't taste like the rest. Next time you've got a hipster style sort of craft beer in front of you, get the light on your phone and put it up against the side of the glass, and then look through the other side of the glass. What you'll see is, you won't see the beam of light coming through, but what you'll see is the pint glowing, sort of similar to what your finger would be doing. So when I talk about imaging brains using light, we're basically just treating brains exactly the same as we would treat a pint of craft beer.
That's a great explanation. I'm going to now have to buy a pint. Thank you for validating my Friday night choices.
I'm very pleased to just make sure it's a cloudy.
Really important. I will go for that. Get myself a cloudy cider, maybe.
I think we can sort of wrap up on that note. I've got one final question. You gave advice for being a science comedian, but if you had any other general advice for our listeners from your science, from your research, from your origin story, from your journey, what would it be?
I think my advice to sort of anyone, and who am I to give advice because I'm not successful, but my advice would be to be interested in things. I remember when I was doing my PhD, and I was thinking, I've always been really, really interested in science, but then when I started my PhD and I started working on a very specific question, I sort of only let myself be interested in that question and in things relevant to that question. And I thought, different bits of science, they're not related to my bit of science. And I remember sort of halfway through my PhD thinking, I didn't get into science just so that I could explore this very niche question, and sort of thinking, no, I can allow myself to be interested in things. I can spend time on different bits of science. I can learn things. And that sort of, and sort of that would be my advice is, yeah, if you're doing a PhD, you're doing something niche, you can be interested in other things. And one of the things that I am really interested in is talking with people about science, and sort of us as scientists, we've got a lot of skills, we know a lot of things, we've learned how to explain things, how to understand things, and so my other bit of advice is just to go out and talk to people about science, because I think it's fun.
That's brilliant. Awesome, everybody. So thanks for that. Thanks again, Liam, for joining. That was great fun.
It's been an honour to hear, and Beth. It's been an absolute honour. I think this is going to go on my CV. And I'm going to reference this in both my Oscar acceptance and Nobel Prize acceptance. I can't even make a joke.
Next time you're in the same lineup with Olivia Rodrigo, you can tell her.
I mean, I didn't. Did I make that joke? Did I make that joke?
You did not make that joke.
Whenever I mention Glastonbury, people are like, "Oh, did you go to Glastonbury, Liam?" And I'm like, "Lol," because I do mention it quite a lot. I got rid of the wristband the other day, and I was showing people that I didn't have the same colour wristband as the rest. I had a performance wristband. But yeah, I was on the same lineup at Glastonbury as Olivia Rodrigo, Charlie XCX. I mean, I've made it. There we go.
You said you weren't successful before. Those are lies.
Look at how successful you are. There
lies.
You go.
No, I mean,
okay.
No, I made it. Well, any other big artists want to get me to open their shows for them, I can say 3 quid in a Welshman. I'm ready for it.
Awesome. So on that note, if anybody wants to hire Liam to open shows for them, you can find him through us. But otherwise, take care. Thank you, everybody, for listening. Thanks again, and until next time. Bye.
Bye.